Hello, good evening all. Welcome to our channel Allied Educare. I am MT Kaushik Sen. On behalf of Allied Educare, today I am going to teach you what is DNA vaccination. So in previous lecture, we have already learned what is vaccination, the invention of vaccination, different type of vaccination, and how vaccine works in our body and how immune system is activated by the vaccination. So in its previous lecture, we saw that there are different type of vaccination. There are different types of vaccines. Let's say uh, live attenuated vaccine, it may be toxoid vaccine, is maybe subunit vaccine, so different type of vaccine. So in all cases, what we are doing, we are extracting the bacterial antigen or whole bacteria itself, and we are administering the virus or bacteria in our body. So in case of live attenuated vaccination, we just eliminate the virulence of the virus or bacteria, and we are administering the bacteria that lost its virulence by vaccination. And after that, the immune system will be activated by the stimulation of the antigen of the virus or bacteria. So in case of this type of bacteria, in case of vaccination, where we are administering the bacterial or viral subunit or total virus itself, there is a problem. What is the problem? It may There is a possibility of a person getting infected with the virus or bacteria. There may be possibility, and sometimes it is observed that this vaccination will not work at all. So DNA vaccination is very safe. Is very safe. So instead of any type of bacterial antigen, bacterial protein, or viral protein, or total bacteria or virus itself, we are using the DNA of the said virus or bacteria. Actually, not in bacteria. We are using the DNA vaccination in case of to protect us from the to the virus. So DNA vaccine is a type of vaccine that used genetically engineered DNA. So let's suppose this is a virus. This is a virus. This virus having a nucleocapsid, and in the nucleocapsid, this virus having DNA. This is the DNA. So not we are just extracting out the DNA. We are extracting out the DNA, but we are not directly administer the DNA into our body. If we do that, there are some problem. So we have to do it differently. We have to genetically modify the DNA first. Then we have to administer the genetically modified DNA into our body by the vaccination. So what is the definition of the DNA vaccination? DNA vaccination is a type of vaccine that use genetically engineered DNA. Actually, the DNA that we are going to extract from the virus, it should be genetically modified. How it is modified? We are using non-replicating circular plasmid. We are using replicating circular plasmid to promote the body cell to produce the specific antibody. So in the recent year 2021, this DNA vaccination is introduced. Actually, the in case of in the endemic, when the COVID-19 is present, so 2021, this vaccination will be introduced. So what are the advantage and disadvantages? So vaccination having some advantage and disadvantage also. So what is the advantages? Advantages mean, as we are not administering the bacterial antigen or viral antigen, so there will be no chance of getting infected. That means no risk of infection. There is no risk of infection from the said bacteria or virus. The immune response focuses on the antigen of interest. If we administer a vaccine, if we administer a virus, there are different type of antigen, different type of protein. So against each protein, the body needs to generate different type of antibodies. So we are just pinpointing a particular type of antigen against which the antibody will be produced in our body. Third point is ease of development and production. It is very easy to development and production. Stability for storage and shipping. The DNA vaccine is much more stable as compared to other type of vaccines, and it is cost effective. So there are 5 advantages, but there are some disadvantages as well. There is a limited to protein immunogen. The vaccination is only limited to protein immunogen. In case of vaccination, that is, we have previously learned that there are different type of vaccination. So what we are doing? We are doing just picking up the antigen and administering in our body. So the antigen may be protein, maybe lipid, maybe carbohydrate also. But in case of DNA vaccination, we can only generate the antibody against protein immunogen, not against the lipid antigen, not against the carbohydrate antigen. But there are some limitation. Why so? I am going to discuss about the next slide. And there is a possibility of cross contamination when manufacturing different type of live vaccine in same facility. There are some cross contamination. So there are two disadvantages. But in recent year there are lots of controversies about DNA vaccination. This is not effective, such blah blah. So we are going to discuss about now how the DNA vaccination is made. So let's suppose it is a virus. This virus having a genetic material. So, this is genetic material. This is the virus. This is the capsule envelope of the virus, and this is the antigen. This is the antigen, and this is the nucleocapsid, and this is the genetic material. Let's suppose it is a RNA. Let's suppose it is a RNA virus. So in case of RNA virus, there are lots of coding region. This is the coding region, and this is the gene responsible for the coding of viral antigen. So this is the coding portion of the RNA that produces at the specific antigen. This antigen is this type of antigen. This is surface antigen. Let's suppose this gene, this specific gene, that is these two gene. The two gene are making specific antigen that is called surface antigen. So we need to generate immunity but through vaccination against this type of surface antigen. So what we need to do? First of all, the process is called extraction. As we learned from the PCR chapter, there is a lots of steps. The first one is extraction. So what we need to do? We need to do the extract out the genetic material from the virus. So in case of first step, we need to extract out the genetic material, and it is genetic material. We have extracted out the genetic material, and now the genetic material should be amplified. How it is amplified? Just look at the next slide. So let's suppose this is the RNA. This is the RNA, and there are two regions. One is coding region and non-coding region. So this coding region is called exon. This is the coding region. This coding region actually making the specific antigen against which we need to generate antibody in our body. And this is the intron part. This is the intron part. So this is the non-functional. This is non-coding part. So we don't need to bother about this part. So what we need to do? We need to develop some primer. Primer means the primer binds a specific nucleotide present in RNA. So we need to generate a specific primer, and in the next step, reverse transcription is taking place. This is a reverse transcriptase enzyme, and this procedure is going on in vitro, not inside the bacteria or inside our body. As we extract out the genetic material, this genetic material is undergone some amplification. So what we are doing? We are making some primer that is specific against the RNA. This is the RNA primer, and RNA primer is binding with the RNA in a specific region, and now the reverse transcriptase will make this strand. This is called complementary strand. It is making complementary strand, and ultimately, as the reverse transcriptase complete its job, then its cDNA is formed. This is the cDNA. cDNA means complementary DNA. So this portion is from the RNA, and this portion is from the DNA. So this is the complementary DNA. Now what we need to do? We need to insert the complementary DNA into a plasmid. Plasmid is a non-replicating DNA. So this is the plasmid. So what we have done? We have done just inserted the cDNA. This is called cDNA. This is cDNA. We have inserted the cDNA into the plasmid. Okay. Now we need to amplify or grow the plasmid that carries the cDNA into a specific bacteria. The plasmid carrying specific gene are grown into large quantity in bacteria. So let's suppose this is the bacteria, and we are injecting the plasmid that carries cDNA into the bacteria, and lots of plasmid will be generated in this way. Now we are going to purify. We are going to collect and purify the plasmid. So how to purify? By column chromatography. Plasmid are purified by column chromatography. So these are the plasmid. These are the plasmid. Plasmid means it is a non-replicating DNA, and we are inserting the cDNA into the plasmid. And after the insertion, we are going to grow the plasmid inside a bacteria, and we are just collecting the plasmid from the bacteria. And after the collection of the plasmid from the bacteria, we are going to purify the plasmid by column chromatography. And after the purification, we are making the vaccination. And as you see from the picture, this is the ampoule of the vaccination or vial of the vaccination, and this is all plasmid that having a cDNA portion. And finally, we are going to inject the vaccine into our body. So as the cDNA that carries as the cDNA enters into our body, it will be transcribed. The transcription is taking place, and from this portion, this is the cDNA. This is the cDNA, and the cDNA is transcribed into mRNA. And this process is called from DNA to RNA. This process is called transcription. The transcription is taking place. And as the mRNA is generated, the mRNA is going through translation in endoplasmic reticulum. So what happened? After the translation, the viral antigen will be produced. This viral antigen. So we are not directly injecting the viral antigen. Instead, we are injecting the genetic material of the virus by different technique in our body, and from the genetic material of the virus that is pre-treated in vitro, and it is inserted into the plasmid. And what happened? This type of cDNA will be transcribed into mRNA, and mRNA is translated into viral antigen. So we are just going to initiate the immunity against the bacteria indirectly, not directly. In case of other vaccination, that is live attenuated vaccination, toxoid vaccination, we are going to inject the antigen itself. But in case of we are making the immune system activated by inserting the genetic material, and finally the genetic material will produce the viral antigen, and against the viral antigen the immune system will be activated and B cell will produce antibody. So this is the summarization of the total process. So here they are taking the West Nile virus as example. So there are two types of gene. There are two types of gene, that is PRM gene and E gene. And this type of gene actually is the RNA virus. This gene is taken out of the cell, and it is inserted into the plasmid, that is a non-functional DNA, and finally it is grown into the bacteria, and after that it will be collected and it will be purified by column chromatography, and finally from the purified plasmid a vaccine is made. So let's look at how the immune system will recognize the antigen and it will produce antibody against the antigen. So as we see that from the picture, the DNA vaccination is administered in our body, and finally by this process By this process, the viral antigen will be produced in our body. So what's happened? As the viral antigen is foreign, so macrophage recognize the antibody antigen, and it will undergoes phagocytosis. It will engulf the total antigen. It is total the engulf the antigen, and the phagolysosome is formed. Phagosome plus lysosome is fused, and phagolysosome is formed. In phagolysosome, the antigen will be degraded, and the antigen will be displayed by the MHC class II molecule. As the macrophage is antigen presenting cell, it has MHC class II molecule on its surface, and the antigen will be displayed by the MHC class II molecule. What happens next? The macrophage migrates towards the lymph node. Lymph node is a secondary lymphoid organ where we found the naive CD4 cell or T helper cell. So now, after that, the antigen presentation takes place. What happened? The macrophage will display the antigen with its MHC class II molecule, and it will be presented to naive T cell. Naive T cell is sleeping mode. Naive T cell is in its sleeping mode. It is not activated yet. As the MHC class II molecule is bind to the TCR receptor or T cell receptor of the CD4 cell, and there are another two types of interaction, that is CD80 ligand and CD20 ligands from the CD4, and there is the interleukin 4 secreted from the macrophage, and these three signal stimulate the T cell, and this T cell gets activated. Once the CD4 cell get activated, it will undergo proliferation, and after the proliferation, a new type of cell is produced, that is called Th2 cell. This is called activated T cell or Th2 cell or T helper cell. This is all about T helper cell. In the meantime, what is happening? B cell also present in lymph node. So B cell also is a antigen presenting cell, and it is having B cell receptor. As a B cell receptor it may be IgG. Sorry, it may be IgM or IgD. There are two types of receptor present on the B cell surface, and as the B cell is a antigen presenting cell, so it must have MHC class II molecule on its surface. So as the vaccination is administered in our body, so viral protein is generated. Some of the viral protein it gets into the lymph node, and B cell receptor will capture the antigen. This is the antigen. This antigen, which type of antigen? This antigen. We are talking about this antigen. This antigen is produced by replication, translation and translation. So translation process, the antigen is produced. Now what happens? B cell will capture the antigen with the B cell receptor, and it will process, and as it is an antigen presenting cell, it will process the antigen and it will display the antigen with the MHC class II molecule that is present on the surface of the B cell. Now from here to there, now what is happening? The B cell is still inactivated condition. B cell is still in inactivated condition or semi-activated condition. It is not activated yet. So what happened? Now B cell will display, as it is antigen presenting cell. As it is antigen presenting cell, it will display its the antigen, the foreign particle. This is the foreign particle. It is a non-cell protein by MHC class II molecule. To whom? To T cell receptor, to Th2 cell. It is representing displaying the antigen to the Th2 cell, and Th2 cell will bind to the MHC class II molecule with T cell receptor. And there are another two types of interaction will be there. There is a first interaction, there is a second interaction, and this is the third interaction. So due to the three types of interaction, this B cell will be stimulated, and after the B cell will be stimulated, the B cell gets activated, and this activated B cell will undergo proliferation, and there are lots of genetical modification in their gene, and finally a plasma cell produced. And this plasma cell, we know that the plasma cell is a antibody making factory. The plasma cell will produce lots of antibody, and this antibody remains in our body, and whenever the virus infect our body, this antibody will binds to the virus, and it will eliminate the virus. And uh, another cell, that is the memory cell, it is also produced during the activation of T cell and B cell. Another cell that is called memory T cell, and a memory B cell also produced. Two types of cells are produced too. So what the function of the memory T cell and memory B cell? Memory T cell and memory B cell just like a memory. The memory T cell or B cell will come into action in very quickly. The memory B cell or T cell will uh, identify the antigen very quickly. So there are less time is taken by the immune system to generate antibody. So this is how the DNA vaccine will work in our body. And in coming days, the DNA vaccination will may replace, not will, it may replace the conventional vaccination method, because it is a safe method and it has less risk of getting infected, and it is very effective and accurate and precise. But there are some controversies about DNA vaccination. So this is all about DNA vaccination. If you have any question, please ask your question in your comment section, and do subscribe, like and share our channel, and obviously follow our channel for the new updates. So thank you very much. Good night all. See you in next class.